So just to briefly introduce myself, most of you here in this room know me. So I'm a field application specialist here. And my aim is, of course, to make all my customers successful and get a good story out of their township profiling assays. And that's what drives me, and I find it very exciting. But of course, yeah, it's always helps to have a structure to it. So we are constantly working from PAMGene side to improve how we report data. And due to my years of experience here, I've sort of accumulated some list of key factors and things to keep in mind while we report our data, because the data can be very complicated, and then giving it a good structure will help. Um, so I will share first, as a first thought, I will share like a report that we've currently sort of evaluated, and that's the current version of it. And then I'll go about telling you how we arrived at this report. So this is more like a sneak peek into what we're going to talk about. So you'll have an idea of what to expect during the seminar. So let me just share the report screen.
New guests are joining. Luxembourg has arrived.
Yes. Hi, Joseph.
Hey, Joseph.
I was just
Welcome.
getting started, to share, the report, the main report example, for instance. Can you
Nice.
see the screen now? Yeah, great. Yeah, so just straight away tell you, okay, this is kind of like a current reporting format that we've developed. This is a very generic example with, some falsified or data or hidden data. So initially there's an introduction, and then we just explain the results. I'll go over more details, of course, during the presentation, and how we do the analysis, what's the kind of QC flags that we give to the data, and also phosphosite at the phosphosite level. I'll explain what each of these are, and then to explain what are the key kinase results. And then we have a supplement. I think I'll have to share that again.
If you like, yeah, you can.
Yes, the supplement. Yeah, the only thing is we have to share one by one, I think.
Yeah. So that gives you a feeling on where we're working
exactly.
towards.
The supplement. And so this is another generic, the same example. So we have an introduction about the PAMGene technology here, which most of you are aware about. So this is not something I'm going to cover. So this is put here. And then we have the data analysis overview. I'm going to talk about this one in a bit more detail. And of course, we have all the different types of processing of the data. What are the quality flags we apply? And the actual results, what are the quality flags and why? Do we apply any kind of normalizations on the data? And then at the phosphosite level, what are the different significant phosphosites in the t-tests? And then these are some of those results. And of course, the kinase analysis, providing the differential upstream kinase data together with some plots and additional tables and trees, because the report we're just listing what are the top kinases that are differential. So this is kind of it. And then, of course, the references and conclusions. And we will discuss this now in more detail at the reporting point.
Yeah.
So I'll share the presentation now.
Maybe it's nice to share that we will upload both of these documents also on the core facility page. So you can all find them there, and you can copy it and change it the way you want to use it. So this is really a resource we want to share with you all.
Yeah. All right. So maybe you could mute your video, Eddie, and then it'll be easier for the presentation. Yeah. So yeah, today I will share with you some of our ideas and experiences. And of course, we always love feedback from our customers also. So please feel free to let us know if there's something additional you think is important or critical in reporting, which I haven't mentioned today. So this was all the previous talk, and then today I'll continue with the reporting part. And you can find back all of these materials like these videos and presentations at our core facility, and also additional information on the general users. So today I'll explain a little bit about understanding of PAMGene data, the critical aspects involved in reporting, the structure, provide some examples that you can add to a report, and then the conclusions and interpretations. So for understanding of PAMGene data, so just to say, as you all know, it's like the tip of the iceberg, and it can be quite challenging because the underlying information is what is very complex, but yet very, very critical. So this is something that we all would like to address in any kinds of omics analysis, and it's part of PAMGene's challenge as well. So just to simplify PAMGene data, it's very important to remember that it's a comparative analysis where we always compare any two given conditions, a test, one or more test conditions versus a control condition. And so when you compare a test and a control, and then you see what are the differential phosphorylations. So as you all know, what spots you see on the array are actually the phosphosites that are being phosphorylated by the kinases in the lysate. And so each signal is actually a collective signal which could result from the phosphorylation by one or more kinases, because each kinase can phosphorylate more than one target on the PAM chip. And so we look for relative differences between these phosphorylations. At the kinase level, of course, we were interested to know what are the different top differentially activated kinases, because every phosphosite, as I said, can be phosphorylated by several kinases and vice versa. Each kinase phosphorylates many sites. So this relationship is complex. Therefore, we have developed a functional tool called the upstream kinase analysis, by which we could actually find out what are the top differentially activated kinases. So this is going for univariate analysis, the phosphosite one, and that looks at per peptide changes, whereas the multivariate one looks at sets of peptides that are changing in the data. In the PAMGene assay, the important factors are that you are actually measuring kinase activity, which is not really the same as expression as you see in protein or Western blots or protein assays. It's also comparative, as I just mentioned, it's relative. So you're not measuring absolute kinase activity, but differential kinase activity. And of course, experimental design considerations are very important. The main reason being that the primary kinase of interest should be active for the perturbation that you're looking at. Sometimes you might be looking too late or too early, or the concentrations that you see may not be effective in inhibiting a kinase of interest. So a lot of these type of factors go into the experimental design, which is therefore important. So any questions up to this point are welcome. Do you see anything in the chat? So
yeah, you can type in the chat if you have a question, or you can also unmute yourself if you like. So if you have but I think at this moment we can continue. So.
Continue. Hey, somebody's waiting.
Yeah, I'm admitting them. Don't worry.
All right, thank you. Yeah. Right. So for just to explain a little bit more about what we consider important. So before any study commences, if you all have had these intake meetings with us, with me or Peter, you'll understand that we do ask a lot of questions during this intake meeting. And that's because we are interested in knowing the type of experiments, whether you're using cell lines or tissues, what kind of inhibitors, what kind of samples, the goals of your experiment. These are very important, like what are you trying to find out? Is it a biomarkers type of study, or is it pathway elucidation study? So these are very important end goals to keep in mind when we analyze the data. And of course, the scientific background information is important about the cellular or animal model. And also we added this at a later stage to know what are the expected steps after this experiment is done, because initially we always give a list of the differentially phosphorylated phosphosites or differentially activated kinases. And how do we go from here to further biological interpretation? To what extent? It's important to set expectations as well, because, for example, the principle of the technology, which I just went over, to explain the actual complexity of the phosphosite to the kinase, and that it's actually a comparative or a relative or differential kinase activity versus an absolute activity. So these are important things to actually explain. And of course, species homologies, because sometimes if you use other species, you may or may not see as many signals, or depends on what kind of model it is. Generally for mouse and rat, we do still see pretty good signals, but it could be lower than the human on which it's based. And what are the main results that you're going to report? What kind of data can actually increase the confidence or trust or transparency of the result? We don't want to just provide these lists of kinases without any clue as to the data behind it. So, of course, the main result is, like I said, the top significant phosphosites or kinases. When it's provided along with the QC data and the QC criteria, then that could be a little bit more robust or acceptable. So when you, some of the considerations in reporting are to talk about study aims, like more detail about the study background and design, because this helps when analyzing and translating the data. Optimal design is very important. As we've sometimes talked about, if you want to run, say, multiple comparisons, it's not a good idea to say run all the controls in one run or the treatments in the other. No one would do that, but I'm just saying it as an extreme example. So these kind of designs would prevent certain statistical tools from being applied, like normalizations, because for certain combat or other corrections, we need to have a balanced design of the experiment. Especially when you're combining multiple runs, this becomes more critical. And like I said earlier, some of the conditions like the time series or the concentrations of the treatments, the controls that you use, these are all very important factors to consider. This is always the question about the number of replicates required. And yeah, we need at least 3 replicates in the case of cells or cell lines, immortalized cell lines, because these are more homogeneous. With biological replicates, when it comes to mouse models or human samples or primary cells, it's better to have at least n equals 6 for each condition or group. So these are necessary for the statistical considerations. Then when you visualize the data, there are simple ways of visualizing it, like you have heat maps and PCA plots. The thing is, we could show everything, but of course, yeah, it's better to avoid showing too many figures that are saying the same thing, of course. Like you could either show a significant heat map or a raw data heat map, but avoid showing both. And it's very important, I think, not just to provide a list of these kinases or peptides, but to give some context, or say a volcano plot. What is a volcano plot, or a PCA plot, or what differences do we see and why? You know, try to add also some biological context to the results.
Yeah, I think at some points they could also talk to us if they have questions about these things, right?
Yeah, yeah, of course. More details of these can also be like provided. And yeah, this is the end of this section. If there are any questions.
Yeah. So if you have ever, you know, come across a question you can't answer, or you want our input, for example, for context or for interpretation help, feel free to reach out to us. Son has a question, I guess.
Yes, I have a question. Exactly. I just saw on your slide that with the homology, there was only written human, and we also came across this with our own samples, because we also have a lot of mouse samples, and we see that the data that we get eventually from the BioNavigator is always based on human genetics. Is there also a possibility to add also, that we can also relate it to the mouse genome?
Beautiful question.
Yes, very good question. Excellent. Thank you very much. Yes, indeed, that is in the pipeline for PAMgene. But it's important to know that the actual sequence is what we blast for our upstream kinase or other results. So we're not really taking the UniProt IDs that are related to these sequences itself. Because these UniProt IDs that we provide, for example, in the array layout and things, these are human UniProt IDs, of course. So each sequence is sort of, each sequence of the peptide of phosphosite is related to a human UniProt ID. But in the upstream kinase analysis application, we're actually starting or originating from the sequence itself. And we also take into consideration the sequence similarity from, say, 40 or 50% up to 100%. So there are options where we, during the analysis, we can change these settings a little bit to make it less stringent for other species. Because we actually we link to databases using the sequence information.
But I think the question also originated, maybe I remember that Astrid asked this question also months back. So Astrid, I think you're happy also with the question from Sanne. But the idea of an upstream kinase analysis for mouse, I think it's a great thing.
Yeah, of course. Yeah, even people in other places in Germany, like Lübeck, have been asking about this.
Oh, yeah.
So definitely, it is in the pipeline, and we are hoping to have it done maybe this year.
Yeah. Okay.
Yeah.
It would be. I think it's a good idea.
Also the chip. So yeah, it is in the pipeline, like I said. But for now, I also explained that the sequence is not just human because it's based on the actual sequence itself. So I think we could develop a mouse or other species. Mouse would be the first one.
Yeah. Just to add on here, but the databases that we use for the upstream kinase analysis are also for humans. So unless we have mouse equivalent databases, that might take a little bit more time to.
Okay. Right. Yeah. So that is fixed databases that we have. Yeah.
So the restriction is not linking the data to a mouse kinome, but the data is originating from kinase to peptide relationships that originate from human experiments. Okay, that may be
from for example, is the phosphonet also extended to mouse, or it's all human derived? I think we have to check that. Yeah. But we are having the ball rolling when we use other databases. If you know some very good mouse post-translational databases, we would love to hear that from you.
Yeah.
Because we also are constantly learning and updating things. So
yeah, I love the initiative. Yeah. So I think it's also on my agenda, but it's good that we bring it up again. Yeah. So
actually, within PAMGene, just to let you know, we have like a team called the Knowledge Integration Task Force, and we are constantly meeting and striving here in this task force to really update databases and things. So this was already one of the projects that we had on our pipeline. So yeah.
All right.
All right. Any other interesting questions?
No, I think you can. Great.
So now talking about the structure. So why did we develop a reporting template? Because we said that, okay, it's nice to create a synergy in the reporting of PAMGene data. Currently, even for within our own self, the services that we report or the reporting to customers ourselves, these we wanted to synergize that. And we, of course, also want to make results self-explaining. So anyone who reads the report should be able to understand it and share it. So right from PhD students to PIs to everyone should understand the report once they read it. It also, of course, leads to a clear understanding of the data of the results because we're explaining everything in the report. There's also a lot of text and background information that is explained. So it leads to a clearer understanding. And we also want to give additional context and explain each graph. Otherwise, in earlier formats when we used to do PowerPoints or something, we can't really explain each graph there, but here we have a legend that explains each graph and result. Of course, one of the most important things is to gain biological insights and for interpretation of the data. So we can kind of point out that, okay, in this particular comparison or treatment, you're seeing an increase in the activity of certain families of kinases, And so once we find the activation of some kinases, we could find ways of confirming this data or to validate the findings and hypothesis. And of course, yeah, it always leads to another set of experiments because, yeah, from this data, where next? And then that can also be discussed in the conclusions. So that gives a extending of the platform usage. There. Yeah, so as I showed you the sneak peek earlier, we have this kind of a reporting workflow that we feel is very clear to really explain the PAMG data. I will go step by step into each of the three of them. And how are each of these steps linked? What is reported in the main report and what's actually included in the supplement that also we provide here in this figure. Just to zoom in a bit on the basic processing. So when we get the raw PAMChip data, which means that the results folder from the evolve, where we just see all images, then of course, yeah, we have to give sample annotations, we have to quantify the images, check the quality of the data, apply normalizations when necessary. And so here we just mentioned what are the normalizations. We report the data quality. Was it fair, bad, or good? And in the supplement, of course, yeah, we provide all this data in the supplement. And from here, we, of course, look at each phosphosite change. And this is called the phosphosite analysis. And this is more to look into the direction and the size of the effect. So is there an overall inhibition, or there's how much is it? Is it very strong? Say between two treatments, how does it compare to control and all of that? And this is mostly based on statistical analysis applied, such as t-tests and ANOVAs. And of course, we can visualize the log4 change here. Most of the time in the visualization, we provide volcano plots and significant heat maps in the supplement, and a list of the most differentially up or down regulated phosphosites on the main report. And at the kinase level, so now we are now looking at how sets of peptides are mapping to a specific kinase derived from our databases. But of course, how this actually changes in your data. So that's what is the functional scoring tool that we have. And so here, at the end result of this is you get a ranked list of kinases, both serine, threonine, and tyrosine, that are actually differentially phosphorylated between any two conditions. So the main report, where we thought it's a nice idea to just have like the top 10 kinases, which is up or down. And in the supplement, we provide further figures like the score table, like I showed you earlier, score plots, the kinome tree, and so on.
So if I can summarize it, we kind of focus a lot of the most important info in the main report and a lot of the side info in the supplement.
Yeah, the visualizations and everything in the supplement. That's right.
Yeah.
Yeah.
Oh, great.
Yeah, great. Thanks for clearing that. So in summary, like just to say, yeah, so we provide like a study specific information like aims and stuff, which is mostly obtained from the customers. And then of course, yeah, the basic results, like I said, the QC data and what are the significant phosphosites, the significant top kinases, and the main conclusions we can derive from it. And like we just said, everything else mostly goes into the supplement with all the details, explaining the technology, the data analysis overview, and also additional visualizations that help you to understand the data better. Here we, of course, have to choose. We sometimes don't have to present everything, but sometimes it could be interesting. So any questions up to now?
Yeah, maybe. I think this is a nice, we share, of course, a slide deck as well. So if you want to read back or, see the list on how we built this report up, you can use the slide deck to have a recap on that. But I think there's no question. So,
but currently, yeah, just to say, like we have a sort of an automated, semi-automated version where we save data tables and figures, and we automatically knit a report. But what we can provide to you is like the template itself for now, and then yeah, you could also add the figures or tables yourself. But at some point when it's ready for sharing with you, then we can also provide you with this kind of a format where maybe you could make a knit a report, right? Like the code or something like that.
Oh, the automation, yeah.
Yeah, for the automation.
Yeah, I wasn't talking about the automation.
I just gave it as additional information.
Okay, great. Yeah.
So these are some example reports, example results in a report. So this is important to understand what we actually report and why. So why do we want to talk about data QC? Because we want to give a feeling on the quality of the data and to show if the data is reliable and interpretable. And the phosphocyte level data is quite necessary to show the trends, like I said, the effect size and the direction. And of course, we always can get a differentially phosphorylated targets and deregulated pathways from this, because often you can move this into pathway analysis software, such as the Metacore, Ingenuity pathways, or many of the free ones like gProfiler, KEGG. There are many pathway analysis to which you can input this differentially phosphorylated data as well to obtain more context. And heat maps can often help in identifying patterns. For example, in biomarker developments, we often see that when you say correlated to a particular factor, like a responder and a non-responder, we see very different patterns in prognostics or diagnostics biomarkers. So the pattern of the phosphorylation heat map could be very important there. Sometimes, yes?
There's also a question from the chat. I don't know if you want to finish the slide first.
Yeah,
and then I'll ask the question. Yeah.
Okay, sure. Yeah.
Okay.
So then there's this third is the kinase level analysis, which is of course the most important result that people expect. And you want to see the differential kinase activity between two conditions. This is also, it could be important in inhibitor screening or profiling, and to identify the key kinase networks in pathway elucidations. Any question?
Yeah. No, it was an interesting question from Emil. That's related to the question we discussed before on the mouse kinome.
Yeah.
Do we have a list of kinases that are included in analysis that are present in human samples, but for example, not in mouse or rats? With such a list, they could then afterwards exclude these samples, or these
exclude these kinases. Yeah.
Yeah, based on the species. So maybe
species specific. Yeah.
Yeah, maybe that's a good idea.
That's a good input. We'll take that into consideration and see how we can use it in our application. Of course. Yeah.
Maybe Peter, any ideas already, or no? No, but it's a good question.
It's a good question, and we definitely like such kind of feedback, because that helps us to perhaps improve the app. Maybe we can add a column saying mouse, human, or something, specifically. So we can think of it. Hippo.
Thanks, Sam.
Okay. The next step was the data QC. We sort of have a flag system. I'm afraid this is in modification at the moment, because ever since we changed the camera system, we have to adjust this a little bit, because the signals they're more sensitive. So you do get higher signals with the new system, but we will modify it. But for currently, we just have arbitrary cutoffs here saying that, if your signal strength is in this range, or your phosphocytes are in this range, and how do we do these QC pass criteria, how many peptides come through it, and what is the replicate CV. So this all just gives a good picture of where your data is. It's not black and white. It doesn't mean if you get a fair QC that you can't rely on the data or interpret it well. So it just depends on the context, of course. Sometimes if your CV is a bit higher of biological replicates, then you might be also questioning that. And then we can have certain normalizations to further minimize this kind of variance in the CV. Sometimes if you have another mouse model, of course, the number of QC pass peptides could be slightly lower. And let's say it's in the borderline. If you say have 122 phosphosites it's still, because this is like a automatically generated table, you might see that it's fair, but if you actually see, okay, it's almost good, So that's why we have to kind of judge this more on a subjective basis.
I think we designed it also to kind of bring up the discussion that some data sets are not as good as the customer might think it is. And then from this flag system, we had kind of a point of discussion like, okay, your data is maybe a bit poor, and therefore you shouldn't emphasize too much on the results or discussions.
How to improve it. Yeah. Perhaps we could increase the number of replicates or change a particular condition, these kind of things. And
yeah.
And that like I said, the pattern-based visualization heat maps, it's obvious. Say there are two conditions, you can see that there are overall differences in the signal intensities of different biological replicates. So it's not just by random chance. And this is better visualized, let's say, in a scaled per row heat map visualization, where you could clearly see that there are distinct differences between the two conditions. We have also the volcano plots, which show the effect size and direction. So these are typically what we show at the phosphosite level data to look at differences between treatment and condition. If it's many treatments, then it becomes a ANOVA type of an analysis. Or if it's just a t-test, then it could be just one volcano plot. So this is quite necessary to understand how the data changes. Then for the kinase level data, these are what's all shown in the supplement, for instance. We could show like if there's just this is a tyrosine kinase example, and you can visualize the coral tree, where we can see which families are the most activated or not, or inhibited. So here, for example, the size of the circles will show the ranking, which are the top ones have bigger circles. The color will show you the direction, either it's an activation or if it's blue, it's an inhibition. And then we also can color. This is a new feature we kind of have, that we can color the top kinases both according to specificity as well as according to family. So in this particular example, it's colored by family, but the ordering is still according to the specificity scores. So we can see that, some of the families are all together differentially regulated. And nice. It's nice to see this view, sort of to see, okay, what are the main families? And when you add also the serine threonine data, then it becomes really interesting to distinguish this. Yeah. Any questions up to this point?
Yeah, I would also want to share that if anyone has an idea for visualization or for, statistical test, or, you could also share that with us and we might learn from that as well. So hello, Astrid.
Hi, hey guys.
Nice to see you again.
Nice to see you too. I'm really excited about this coral tree. So, I'm just preparing the trees with the KinMap tool. So which is the tool where you could, highlight the different kinase classes? So from your previous slide, maybe I missed it. With how this can be done by online based or in the BioNavigator?
Yes. So you're asking about this coplot, Astrid, if I'm right?
The coral, this coral tree the colorful tree.
Right. So that comes actually from the data that you download.
Yeah.
We have sort of a way where we can automatically plot this by bringing it into an RStudio format, or go directly in the website. I can provide you extra instructions on how to do that, and then just manually copy paste the values from your statistic or final score, and then it gets projected on this, and it's fully customizable, where you can change the colors or you can change the sizes you can change the legends, and that way really customize this map. And also the nice thing about this is that it has links. So the SVG version, which you download, for example, if you press on any one of the kinases, it links it to the UniProt database.
Nice.
So it's kind of, it's very handy. And you have other views if you actually do this online in the Coral Tree app. It's also possible to have other visualizations, like a force-directed view, not just the tree view. On the left, the score plot coloring by family is that we have now an updated upstream kinase analysis app that we haven't yet. It's still a little bit in the testing phase. We'll soon provide it on the BioNavigator site as well. So this has the possibility to have 2 extra columns which indicate the group and the family.
This is great. This would be very, very helpful. Yeah.
Yeah, so then you can also color it.
Good to know. Yeah, thanks.
All right.
I just put the link to the Coral application in the chat.
Okay. Right.
Thank you. Cool. Thanks.
But yeah, if you need help with it, we can also do an online session to share that, right?
Thanks, Astrid.
Thanks, Peter.
Nice.
For the link. All right. Nice. Wonderful. Great. So then we come to the last part, the difficult part, I would say, the conclusions and the interpretations. And so, of course, yeah, there are all kinds of discussion of results that you have to do. Like, for example, is the replicant variance high? And so would then you answer the question, maybe if you repeat it with more number of biological replicates or you improve sample prep, then you might or maybe make it more uniform. There are many different options or remeasure protein amount. So there are many things for reducing the replicate variance as well. Are the treatment conditions informative enough? Perhaps you might want to add new time points or different concentrations that could improve the results. Or is the data QC reliable? So what are different ways how we can actually improve the QC? Like again, bringing into consideration uniformity of sample prep, protein measurements. There are many reasons, many different ways we could have to improve this. And then often the question is, are the results actually significant? I mean, so we do provide a list of the top phosphosites and kinases that pass a certain threshold. So in that sense, they are significant. And the ones that don't pass the threshold, we're generally not sharing that. But these thresholds are, of course, user defined. So you can either make it more or less stringent. And have we managed to have a pathway and network context to the result? Does it fit well with the current expectations? This brings us more to the interpretation part of it. So does it fit? Like, does the experiment satisfy the aims of the project? Did they already have a hypothesis? Is that satisfied? Can you improve the experimental design further? Is the basic hypothesis validated or verified? Can we corroborate PAMChip data to other findings? Like often people already have RNA-seq data, proteomics data, or just to literature or the current knowledge, say MAP kinase is activated, and do we also see that? And how can the data be then validated? So you do see certain kinases are activated. So in the past, of course, yeah, we have a lot of publications on our website where PAMChip data has been validated. For example, in Western blots or in vivo in vitro experiments, by knockout experiments, inhibitor studies, and also clinical data sometimes has shown validation of a particular finding. So there are many ways to do that, but it's important to at least discuss that in our conclusions. Because the upstream kinase is a hypothesis generator and we have to validate the data.